Hello and welcome to Nikolay's genetics lessons, and today's question is: which of the following statements are true about protein structure? And statement A: all proteins have a quaternary structure. Take a look at this picture and you would see that hemoglobin has quaternary structure, but myoglobin doesn't have quaternary structure, so many proteins just have tertiary structure, but some has quaternary structure, meaning that many different polypeptides aggregate in order to make one functional protein. Sometimes such subunits can be identical polypeptide chains, like in this protein in hemoglobin. Two alpha subunits are identical. Two beta subunits are also identical, but alpha and beta are different, and these polypeptide chains are coded by different genes. So first statement is false. Now let's check second statement. Disulfide bridges primary maintain secondary structures as opposed to tertiary. Secondary structures are made by hydrogen bonding and also hydrophobic and hydrophilic interactions, and those disulfide bridges also can stabilize secondary structures, but the main role in stabilization of the tertiary and quaternary structures of the proteins. Next statement: pH does not change the charge of the protein. This is also not true. Take a look at this table. We see here 8 amino acids, and 2 of them, aspartic acid and glutamic acid, has negative charge in the R group, but this negative charge in the low pH can be lost, and these R groups can be neutral. And for example, these 3 amino acids here, lysine, arginine, and histidine, have positive charge, but again they can lose this positive charge in the R group if, for example, pH is going to be higher, and then they're going to lose one hydrogen in the R group and would be neutral. In histidine R group you do not see positive charge, just like in arginine and lysine, because it gets positive charge at lower than physiological pH. But its R group also can get a positive charge. So as you see, pH plays very significant role in the changing of the charge of the protein, so this statement is also not true. And next statement: a protein function depends on its spatial conformation, and this is true. For example, take a look at the hemoglobin or myoglobin. Its function depends on its spatial conformation and also active sites. In this example, this is going to be heme group in every polypeptide chain. So as you see, the correct answer is D, and this is all for today. Subscribe and see you in the next video. Goodbye.